All right, so let's talk a little bit about how peptide hormones are made. Okay, so first question, this is a question on your worksheet. I want you to rack your brain, see if you can remember, how do our cells make proteins, right? So what is the process by which our cells make proteins in maybe like a sentence or two? So go ahead, hit that pause button to answer the question. Okay, hopefully by digging into your memory, you were able to remember that proteins are encoded by DNA, right? So that DNA is read and we create an mRNA molecule, like a copy of the DNA through a process called transcription. We then read that mRNA and use that to make an amino acid chain, a peptide chain, or multiple peptide chains, which then can combine to form a protein. And that process is called translation. Okay, so hopefully you remember that from bio. If not, you just got your little review. You also might remember that for most proteins, this process happens in the cytosol. Okay, but for proteins that are going to become membrane proteins or proteins that are going to be secreted, such as hormones, okay, this process takes place in the organelle system of the cell, so the ER and the Golgi. Okay, so let's take a look at that. Okay, so you might remember the term secretory pathway. Hormones are made through the secretory pathway. So this is what we are going to be illustrating here is the secretory pathway. Okay, so we're going to take a look at how peptide hormones are made and secreted from the cell, starting with this diagram here. Okay, so first I'm going to just orient you to this diagram. Okay, first we're looking at the inside of a cell. We've got the ER here. We're moving on to the Golgi down here. Okay, but the thing that I really want to draw your attention to is these sort of ribbony, snaky looking things right here, right? You can see that you've got multiple colors in these little ribbons. Okay. This whole ribbon represents the molecule that's going to become the functional hormone, okay? However, it's just the blue part of this little string ribbon that is going to become the actual end product, the actual functional version of the hormone, okay? So let's take a look at what these other things are. And what happens to them during the processing of the hormone, okay? So you might remember that proteins that are going to be secreted or that are going to become membrane proteins, they all have a sequence of amino acids at their end terminus, right? At the sort of beginning end, the beginning. And that is called a signal sequence, right? So what happens is we have translation happening in the cytosol. Once the signal sequence gets translated, okay, the signal sequence is shown in red here. Once that signal sequence gets translated, it signals the ribosome to bring this whole complex over to the ER, to the endoplasmic reticulum, right? So we have our protein that's in process of being translated. The ribosome sees a signal sequence and it says, oh, this protein is going to be either a membrane protein or it's going to get secreted. So I need to bring this whole complex of the mRNA and the ribosome and the peptide that's being translated to the ER. Okay, so that's what happens. Is the ribosome comes over to the ER. The actual process of translation happens kind of across the membrane of the ER so that the polypeptide chain that gets made ends up inside the ER. Okay, so that's what we're seeing here. Okay. After that happens, we no longer need the signal sequence, right? The signal sequence had the sole purpose of directing the ribosome to the ER, okay? So we're gonna cut off the signal sequence right here, okay? Oh, I forgot, I need to introduce these terms, right? So the entire polypeptide chain, including the signal sequence, is referred to as a pre-pro hormone, okay? Once we take that signal sequence off, we end up with this, which we refer to as the prohormone, right? So this is still not the functional version of the hormone. Remember that the functional final version of the hormone is going to be this blue part right here, but it's also got this extra stuff still on it. So all of that we refer to collectively as the prohormone, okay? All right, so that prohormone is going to be processed. It's going to go through the Golgi complex. You can see the label right here. Okay, it's going to go through, it's going to go through, and then finally, it's going to end up in a secretory vesicle. Okay, that secretory vesicle is going to wait for a signal from the outside to tell it to fuse with the membrane in order to secrete that hormone out into the extracellular fluid. Okay, but as that molecule waits in the secretory vesicle, it actually gets cleaved into the active version of the hormone, which again is this blue part right here, and these other fragments, which are shown in magenta, these peptide fragments, okay? And once that secretory vesicle fuses with the plasma membrane, we then release both the active hormone and these peptide fragments out into the extracellular fluid, sorry. Okay. So here are some examples of peptide hormones in various stages during this process, right? So here's an example of a pre-pro hormone. It's called pre-pro TRH. TRH is thyrotropin releasing hormone. And as you can see, this pre-pro hormone has this signal sequence. It's got these little blue sections, okay, which are then going to become the actual functional version of TRH. And then it's got these little pink portions, which are those peptide fragments that are not functional, okay? So in this particular case, we have one amino acid chain, one polypeptide chain, that's going to give rise to 6 copies of TRH, okay? Looking at this example, this is a prohormone called pro-opiomelanocortin, or POMC, okay? Remember that a prohormone is missing the signal sequence, right? So previous to what we are looking at here, we had a red portion up here that already got cut off, okay? POMC consists of these different parts that are going to be cut apart to give rise to a hormone called ACTH, adrenocorticotropic hormone, and then a couple other functional molecules, okay? And a non-functional peptide fragment, okay? Our final example here is insulin, okay? So what we're looking at here is proinsulin, right? So we had our polypeptide chain, which actually, in this case, folds on itself and is held together by disulfide bridges, which you may or may not remember from your bio classes, right? So it's held together by these disulfide bridges. After it goes through processing, we end up with functional insulin and this non-functional peptide called C-peptide. And sort of an interesting side note is that C-peptide is actually used as a test, like levels of C-peptide are used to test for insulin levels in a person. So they're used as a proxy for insulin levels.